Hi everyone. This week we're going to be talking about energy, and specifically how energy is going to affect living things. Next week we'll be talking about how that energy is generated inside of the cell, but this week we're just going to be talking about the energy of chemistry and the energy of life. We've talked before about chemical reactions back in week 2, and in every cell in your body, there are thousands of chemical reactions going on all the time. Those chemical reactions can go in lots of different ways, but they're all happening simultaneously with one another. And it is only by virtue of these things happening that you continue to stay alive. We have a term for all of these chemical reactions happening inside of your cell all the time, and this term is metabolism. Now, metabolism in biology means something slightly different than you probably think of when you think of metabolism in everyday use. Usually we say, oh, if he has a faster metabolism or a slower metabolism, what we mean is how fast you burn energy, how fast you use energy. In biology, that's only half of what metabolism means. Metabolism means all of the chemical reactions that happen inside of you. Some of those chemical reactions take big things and break them down into small things and get energy out of that process, much like we think of as, burning calories. That's part of metabolism. But there's the other part of metabolism too. We can take small things and then use energy to build those small things together into big things. This is also metabolism. So when you burn glucose and carbohydrates to get ATP, that's metabolism. But when you take that ATP and a bunch of amino acids and use them to build a big, useful protein, that's also metabolism. In your body, in your cells, your metabolism is organized into what we call metabolic pathways. We don't usually do all of the reactions to go from the thing that we start with to the thing that we finish with all at once. Right? There are lots of reasons why we don't do that. So an example of that would be if you take a block of wood and you set it on fire. Right? We're taking this big, complex carbohydrate, wood, which is cellulose, which is made out of glucose, and we're setting it on fire. We're turning it into water, carbon dioxide, and heat, all in one step. Right? So we're just going from start to finish, boom. Right? And boom is the operative term because it makes a big fire. That's not something you really want to have happening in your body. We talk about burning calories, but you don't actually want to set them on fire. You want to release that energy in a controlled way, one step at a time. You do this, first off, so that you don't have a catastrophic explosion that blows up your body, which is bad. But more importantly, because it's much, much more efficient. You can capture that energy a lot better if you bleed it out just a little bit at a time. So almost all reactions that happen in your body are multi-step processes. You start out with what we call the reagent or the precursor or whatever it is you're starting with, the substrate. That's A. And then you have one enzyme that turns it into something else, B. This is what we call an intermediate, because we're not going to stop at B, but you got to go through it. Then you have another enzyme that converts it into C, and then you have another enzyme that converts it into D, and then D is your final product. And so you have these highways, these pathways inside of the cell, each of which has to happen in a particular order, and there's often a different enzyme that will do each part of it. These metabolic pathways can be pathways that take big, complex molecules and break them down and suck the energy out of them. If they are, they're what we call catabolic pathways. Most of what we talk about next week are going to be catabolic pathways, where we take complex things like sugar and break it down into water and carbon dioxide, and we take some energy away from it when we do that. But that's not all of the metabolic pathways that are out there. That's just half of them. The other half is what we call anabolic pathways. Anabolic pathways use energy and take that energy and take small molecules, what are called precursor molecules, and combine them together to form big complex molecules. So when you make a protein out of amino acids, that's anabolism. When you make a new strand of DNA before your cell divides, that's anabolism. Generally, catabolism and anabolism inside of a cell need to be balanced. In order to use energy, you first have to make that energy. We've been talking a lot about energy, but we haven't really defined the term. Because in science, energy means something a little bit more complicated than it often does in normal everyday speech. There are lots of different types of energy. Right? Energy is basically the ability to cause change in the environment around you. Specifically, the ability to do work in the environment around you. And it can exist in lots of different forms. So there's kinetic energy, which is things moving around. There's heat energy, which is things moving around at a really, really, really small level. So what heat actually is molecules moving in random directions. And the more the molecules are moving around in random directions, the hotter something is. We also have potential energy, which is energy that's not currently being used, but which is still possessed by an object by virtue of its position or its structure. So we often think about gravitational potential energy, which is if you take a textbook and you put it on the top shelf of your bookshelf, it has a lot of potential energy there. If it falls off of the bookshelf and goes thunk against the floor, it now has less potential energy. It's used up some of that potential energy in the fall. Another form of potential energy is what's called chemical energy. This is potential energy that's stored in the bonds of a molecule. If you break those bonds and rearrange that molecule from a less stable form to a more stable form, then you are releasing that chemical energy and it can do something. Each of these forms of energy can be converted to other forms of energy, although some conversions are easier than others. This branch of science that studies energy in chemistry, how energy is transferred from one thing to another, is called thermodynamics. And in thermodynamics, we talk about closed systems and open systems. A system is anywhere that energy can be transferred. It's any group of things that energy is going to be transferred between. A closed system is one where energy doesn't come in, energy doesn't leave. The amount of energy in the system stays exactly the same all the time. Closed systems don't actually exist, other than the universe itself, which we hope is a closed system. But, in real life, no system's actually closed, but it makes the math easier sometimes. So sometimes we model something as a closed system. But living things in biology are always open systems. Open systems are ones in which energy can come in and energy can leave. In thermodynamics, we have laws that govern how that energy is moved around. Okay? And the first law of thermodynamics is sometimes called the law of conservation of energy, and what it states is that in the universe, the total amount of energy is constant. Basically, it says the universe is a closed system. Energy can't be created. No new energy is happening. Energy can't be destroyed. No energy ever goes away in the universe. It can leave other things, but it doesn't leave the universe. But energy can be moved from one thing to another, so long as they're both part of the universe, and it can be changed from one type of energy into another type of energy. That leads us to the second law. Not all types of energy are created equal. Some forms of energy are more useful than others. So take a textbook on the top shelf of a bookshelf, right? It's sitting up there, it has a lot of potential energy. That energy could be used to do a lot of different things. But let's say that the book falls off the bookshelf and goes thunk against the ground. Well, it now doesn't have that energy. It's lost that potential energy. Where did it go? It didn't leave the universe. It went into the molecules of the air. So if you're standing close to it as it falls, you'll feel a slight breeze, and then when it hits the ground, it goes thunk, and sound is basically a rapid movement of air molecules. So we've changed our potential energy of the book, which could be used to do lots of things, to energy of the motion of air in the form of sound, which once the sound fades, results in a very, very slight increase in the temperature of the walls of the room. You're not getting that energy back. It still exists, but you can't put it back in that book. There's no putting the thunk back in the book. There's no putting the ring back in the bell. The energy's still there, but it's in a less useful form. It has become more chaotic, more entropic. The second law of thermodynamics states that whenever energy is transferred or changed, some of that energy is permanently changed from useful energy into less useful energy. We call that less useful energy waste energy, and it typically manifests as heat. So let's take a look at the form of the laws of thermodynamics in action here in the form of this bear and this fish. You see here this bear with this fish? The bear is eating the fish. The fish is, even a dead fish like that, is a well-ordered structure. It has lots of very, very useful potential energy. The bear eats the fish and turns that energy into ATP, which it uses to run around a whole lot. The second law of thermodynamics states that any time energy is changed or transformed or used, some of it becomes permanently less useful. So let's examine what happens to the energy of the fish. Well, it changes from this extremely well-ordered, useful structure, like a fish. You could eat a fish and do lots of different things with eating a fish. Once it comes out, though, it's basically carbon dioxide, water, and heat that the bear radiates when he's running around. It's very difficult to take heat, carbon dioxide, and water and turn it back into a fish. It's about as difficult as putting the ring back in a bell, if not more so. So, which of these statements is representative of the second law of thermodynamics? I'm going to ask Josh to pause the video right now and read off these options to the class and get your input. So, of these answers, D, cells requiring constant input of energy to maintain their high level of organization, is consistent of the second law of thermodynamics. If cells did not receive a constant input of energy, if they were a closed system, then over time, as they perform chemical reactions, they would become more and more and more disordered until eventually they weren't alive anymore. So that's why you need to get energy in. It's to maintain your own ordered structure so that you kind of off-put the chaos you're causing onto the rest of the universe instead of keeping it for yourself. So when we're talking about energy in biological systems, we're typically talking about what is called free energy. Free energy is energy that does work when temperature and pressure are uniform. Yeah, you can do work that causes temperature to change. That would be heat energy. Free energy is not typically heat energy. You could do energy that causes pressure to change. A lot of, machines, like your car does this. When you burn gas in your car, it causes a pressure change that causes the pistons to go up, and that turns the wheels. That is a different sort of energy. That's not free energy. But cells usually maintain a steady temperature, and they typically don't undergo rapid pressure changes. So the energy changes that are relevant to biology are typically free energy changes. The term that we use in science for free energy, or the letter that we like to use letters for these things, is G. You can't really measure the total energy of something. It's nearly impossible to. So what we're going to care about is the change in G. How much free energy did you have at the start? How much free energy did you have at the end? When we talk about the change of something in science, what we use to denote that is a capital Greek D, which is this little triangle-y thing, and it's called delta. So delta G is the change in free energy. If your free energy increases, if you have more energy at the end than you did at the start, then your delta G, your change in G, is positive. You gained G. If you lose energy, you have less energy at the end than you did at the start, then your change in free energy is negative. Now this equation here, we don't have very much math in this class, but there is just a tad bit, relates free energy, G, with total energy, which is called enthalpy, H, temperature, and entropy. So we said that temperature and pressure are staying the same in a free energy reaction. So note that the T doesn't have a delta behind it because it's the same at the beginning and the end. If your delta G is negative and your total energy doesn't change, then that means that your delta S, your change in entropy, has to be negative. S is entropy, and entropy is a little bit of a complicated concept. It isn't just disorder. It's the number of positions that a system can exist in. So I had a teacher once tell me that, entropy increases over time, and you can see this because over time your room becomes more and more messy. That isn't exactly what entropy is. It's a nice way to think about it, but it's not really true. Because both a clean room and a messy room have the same amount of stuff in them. And what really matters is how many different potential states a system could exist in. If you have one object, there's a limited number of different ways it could be. It could be faced up, it could be faced down, it could be faced to the left, it could be faced to the right. That's about it. It can be faced in different directions. But if you take that one object and cut it in half, well, now you got two objects, and they could be oriented in a lot of different ways to one another. You could have, the left half be really far away, or it could be really close. The left half could move up, and the right half could move down. The left half could actually be to the right of the right half. And so now there's a huge number more of different potential ways you could arrange these two things. And the more things that you have, the more stuff that you have in a system, the more entropy that it has. So when you take one thing and you turn it into 2 or 3 or 4 things, your entropy is increasing. Now, when we talk about the change in free energy, it can be positive or negative. If free energy change is negative, if delta G is negative, if you are losing energy, we say that this process is spontaneous. So any chemical reaction which is spontaneous is also called exergonic, which means energy releasing, and spontaneous sort of means that it will occur by itself. So in this reaction, we have a ball at the top of a hill, right? So the energy of the ball at the top of the hill is more than the energy of the ball at the bottom of the hill. So the ball rolling down the hill has a negative free energy change. Energy is released, and this reaction is spontaneous. You don't need a whole lot of help to get that ball to roll down the hill. You put a ball at the top of a hill, it's pretty much going to roll down on its own. A reaction which is non-spontaneous is one that is endergonic, or energy consuming. This won't happen unless you put energy in. So this ball's at the bottom of a hill. The free energy change is positive. This ball won't roll up that hill on its own. But if you put enough energy into the ball, you can get it to roll up the hill. But you got to put energy in to get it to happen. It doesn't happen spontaneously. Right, so here's where it gets a little bit complicated, because spontaneous doesn't mean what you think it means. So here I have a ball on a hill. It's definitely a negative free energy change for this ball to go down to the bottom of the hill. The bottom of the hill is definitely lower than where the ball is now. So the delta G is negative. The ball rolling down this hill is a spontaneous reaction. Unfortunately, if we were to have a ball in this situation, it would never roll down this hill. Because while the initial state is definitely higher than the final state, the pathway that it has to take has a higher intermediate state. In this case, this reaction has what we call activation energy, or delta G double dagger. That sort of line with two lines through it thing, that's called double dagger. That's the energy that you have to put in order to cause the spontaneous reaction to happen. And I know it's weird to think that you have to do something to cause a spontaneous reaction to happen. This reaction is spontaneous, according to thermodynamics, but it won't happen by itself, because it doesn't have enough power to go through the activation energy. It won't happen by itself unless the activation energy is supplied. So see, that ball just isn't rolling up that hill. Now, I could strap a rocket to it again, and that'd probably make it roll down that hill. That'd get up and over the hill. That's kind of overkill in this situation, because it is a negative delta G. It is a spontaneous exergonic energy releasing reaction. This is the situation that enzymes really help with. Enzymes don't change the thermodynamics of a reaction. They don't make an endergonic reaction exergonic. They don't take a reaction that consumes energy and make it release energy. They don't take a non-spontaneous reaction and make it spontaneous. They don't do anything about where the ball starts and where the ball ends up. What they do is they change the path. They lower the activation energy to the point where the ball can just roll down the hill. So I've put together a list of YouTube videos. If you go to my YouTube feed, which I think I give you the information for, if you go to YouTube and you search for Joshua Stomel, you'll probably find it. I'm the one with a bunch of biology videos. I have a playlist called Enzymes. You may want to take a look at those videos. So when we talk about free energy and energy changes are happening all the time. A spontaneous change, in a spontaneous change, free energy is decreased. So the object loses energy and it becomes more stable. A ball at the top of a hill is not a stable situation. That ball's eventually going to roll down that hill. That situation's unstable. Ball at the bottom of a hill is pretty stable. You can walk a right way for a while and be pretty sure that when you come back, the ball will still be at the bottom of the hill. So when you lose energy, stability increases. A system is in equilibrium when it has the most stability it can get. Any process is spontaneous if it's moving towards an equilibrium situation. So, I'm not going to go through all of these, but let's take an example of diffusion. We talked about diffusion before. If you take a drop of food coloring and you drop it into a glass, it's going to start out with a very concentrated, color right where you dropped it. You come back later over time, it will have diffused out throughout the entire glass. Diffusion is a spontaneous process, and equilibrium is reached when the color is uniformly spread throughout its container. So as long as you're moving towards having the dye spread throughout the entire container, that happens spontaneously. Getting it all back into a little ball that you can suck out with an eyedropper, that's non-spontaneous. It will never happen by itself. It can happen if you put energy into it, run that thing through a really powerful filter, and you can separate that dye back out of the water. But you're going to have to use energy to do it. So, as I said, in metabolism you have two types of reactions. Exergonic reactions, which are spontaneous and which release energy. These are typically catabolic reactions. You have endergonic reactions that absorb energy and are non-spontaneous. These are usually anabolic reactions. Here we have the two different types of reactions. The top one releases energy. You're usually taking big things, breaking them up into small things, and capturing that energy as it's released, versus endergonic reactions, where you use energy, consume energy, store it in the chemical bonds of a complicated, useful molecule like a protein or a strand of DNA. If this system were closed, it would eventually reach equilibrium. You would eventually reach the point where all of your energy had been converted to its most stable form. So you see this water wheel here at the top. It starts off with a bunch of energy, and over time, the two levels of water even out. In the second place here, where the water levels are even, in that case, stability has been reached, equilibrium has been reached, and work can no longer be performed. If we were a closed system, that would eventually happen. We're an open system, case B here, where energy is always leaving in the form of waste heat out of the bottom, and new energy is always coming in, for us, typically in the form of food. So in that case, we always maintain a difference in our water levels, not literally, but figuratively in this picture, and that allows us to always never actually attain equilibrium. If we ever attain equilibrium, we die, because no more chemistry can happen. In cells, the most common material that we use as an energy to perform chemical reactions is ATP. Cells do lots of different work. They do chemical work, taking things and changing the chemistry. They do transport work. We talked about that a lot with membranes. And, you transport things. Active transport requires energy. And you do mechanical work. At a cellular level, mechanical work is pretty subtle, but you can see it in that, you can lift at your arm and flex your muscles, right? So that's a chemical change driving a mechanical change. You actually moving. So how do we use ATP to do these things? When I first learned about ATP, I thought, well, is it like the ATP, like the phosphate pops off and there's sort of a laser beam shoots out of it and zaps the whatever chemical reaction is happening? That's how the energy gets there. Or does it heat up the water around it and that's how the energy gets there? So how does this work actually? And the way it works is a couple of different ways it works. Energetically, thermodynamically, the way it works is by energy coupling. An enzyme can take two chemical reactions with different energetics, and by making them both happen at the same time, and that they can only happen if they happen together, it can take the energetics of the net reaction, of both of the reactions, and use that to drive it forwards. So ATP, adenosine triphosphate, is the energy currency of the cell. When you use energy, 9 out of 10 times, this is what you're using. It's actually, even though we talk about ATP all the time, it's a type of RNA. It's a ribonucleic acid. It has a ribose, sugar, adenine. The A in ATP is the same A in the ATGC of the DNA code that we'll talk about in a few weeks. And it has 3 phosphate groups. So here we have the structure of ATP. You got our sugar, they're in the middle, the green bit. We have our base, that's the orange thing. It's a big base, so it's a purine. And we have 3 phosphates there in yellow. When you use the energy from ATP, what you're usually doing is breaking that 3rd phosphate bond. And that's the highest energy bond in the molecule. So let's say we have 2 chemical reactions. In the first chemical reaction, we have ATP being turned into ADP, adenosine diphosphate, and inorganic phosphate. So we're losing that phosphate. That's a very exergonic reaction. It's very spontaneous. The delta G is very negative. A ball at the top of that hill wants to roll to the bottom really, really bad. Let's say we have a second chemical reaction. We're taking chemical A, and we want to turn it into chemical B. It's an endergonic reaction. A ball at the bottom of that hill isn't going to want to roll up that hill. Not very much. But the chemical A to chemical B is less endergonic. It's less positive than ATP to ADP is negative. So we see the ATP curve is much taller than our chemical A, chemical B curve is. So we have an enzyme that can take these 2 reactions and turn them into one reaction. The way this works energetically is that if you take the 2 energy plots and put them next to each other, and you make a 3rd line, which is made by adding the values of each of the individual lines, we have a new net reaction, which is ATP and chemical A turning into ADP, inorganic phosphate, and chemical B. This reaction in net, in total, is now negative. It's not as negative as the ATP by itself would be, but it's still negative, so it can happen spontaneously. That's the energetics of how this works. But in the biology, the biochemistry of how it works is a little bit more complicated than that. So what we're doing is we're removing a phosphate. And when we take this phosphate from ATP, because ATP is such a high energy molecule, it can basically attach that phosphate to anything it wants to. So the release of free energy comes from the ATP either just letting the phosphate go or attaching the phosphate to something else. It's very easy to get ATP to attach a phosphate to something else. So in the cell, we're powering all of these different types of work, mostly by the hydrolysis of ATP. So ATP hydrolysis can be coupled to any other endergonic reaction or many other endergonic reactions, and the net will be negative and exergonic. So we can take glutamic acid plus ammonia and putting them together. That's a positive reaction. It doesn't happen spontaneously. But if you take glutamic acid and you attach a phosphate to it, and ATP can attach phosphates to stuff like nobody's business, well, glutamic acid phosphate, it's much easier to get the ammonia attached to it if that ammonia is displacing a phosphate than if it's just solidly attaching. So taking this glutamic acid phosphate and attaching an ammonia to it, that's negative. So now it's a two-step reaction. The first is you put the phosphate on the glutamic acid. The second is you use that phosphate leaving to allow the ammonia to come in. These two reactions together add up to a negative reaction because you're coupling ATP hydrolysis to the fusion of glutamic acid and ammonia. Often, as we just saw, this happens through a phosphorylated intermediate. ATP attaches its phosphate to something else, and then that something else loses that phosphate as a part of a second reaction. We saw this last week when we talked about the sodium-potassium ATP pump. That when ATP attaches the phosphate to one side of the pump, it opens in one direction, and then when that phosphate goes away, when it gets released to water, it changes shape and it actually allows the molecule to face the other direction, which is what pumps the sodium to the other side. In our little walking vesicle transporting motor protein, we have the same sort of thing going on. So attaching the phosphate from the ATP to the molecule is what causes the foot to lift, and then cutting that phosphate off is what causes it to set its foot down. So every time it takes a step, when it lifts up, the ATP sticks a phosphate on it, and then when it goes down, that's because the foot gets that phosphate cut off of it. If you're using ATP to do all of this stuff in your cell, that means you've got to have some way of getting more ATP. We're going to talk a lot more about that next week. But suffice it to say that it's, I don't want to say very easy, but the ATP-ADP reaction is very, very reversible. And as long as you have enough total adenosine in your cell and enough total phosphate in your cell and enough energy coming in from someplace, food, sunlight, whatever, you can keep putting that phosphate back on the ATP. It just takes energy to do that. So we have this sort of revolving door thing. You make ATP, you use it, and then you make more ATP. If you were to lose the ability to make new ATP, you would be dead in seconds. In fact, cyanide, one of the more potent poisons out there, kills you in 3 steps, usually. Like if you have a big dose of cyanide, you get about 3 steps before you die. The way it works is by stopping your cell's ability to make more ATP. So here's a question for you. Why is ATP an important molecule in metabolism? Go ahead and pause the video and get some feedback from the class about why you think this is. So most of these are true statements, but they don't all answer the question. So this was kind of a tricky one. So the hydrolysis provides an input of free energy for exergonic reactions. Technically, but it isn't like ATP gets hydrolyzed and then like energy ray zaps out and zaps the exergonic reaction. And actually, if it's an exergonic reaction, you don't need to put energy in. You would need to put energy into an endergonic reaction. If its terminal phosphate group contains a strong covalent bond that when hydrolyzed releases free energy, that's true, but that's not what makes it important in metabolism. Terminal phosphate bond has a higher energy than the other two, slightly but not significantly higher. It's the phosphate-phosphate bond, so the second phosphate also has a powerful bond. So that's not the big reason. It's one of the 4 building blocks of DNA synthesis. It's technically a building block of RNA synthesis, but either way, that's not why it's the most important reaction in metabolism. It's because it couples endergonic and exergonic reactions. So we talked a little bit about enzymes and how enzymes speed up reactions by lowering the activation energy barrier. Enzymes are catalysts. They're typically protein catalysts. And a catalyst is an agent that speeds up a reaction but is not consumed by the reaction. At the end of the reaction, the catalyst is the same. Catalysts don't change the energetics, they just change the speed. They allow a reaction which would normally not happen to happen much, much faster. So an example of this is the hydrolysis of sucrose. Sucrose is better known as table sugar. And breaking sucrose into glucose and fructose is a spontaneous reaction. Right? Energy is released in this reaction, not much of it, but some of it is. But the activation energy, delta G double dagger, is high enough that this happens very, very slowly. Like, so slow that, if you take some sugar and some table sugar and you put it in water and just leave it like that, maybe less than 1% of it will have spontaneously degraded into glucose and fructose if you come back in a week. That's not fast enough for your body. Enzymes can make this happen much, much faster by lowering the activation energy required to get it to happen. The way enzymes do this is usually by binding to and stabilizing the transition state, the middle bit. Usually there's a part in the middle which is harder to get through than either the initial or final state. Enzymes kind of make that transition state feel less bad. They make it less unstable and thus lower, here we are, lower the activation energy by stabilizing the transition state. So here's the last question for you. Reactants capable of interacting to form products in a chemical reaction must first overcome the thermodynamic barrier known as the reaction's what? First off, let's assume that this is a spontaneous reaction. Okay? What is it that you have to overcome in order to allow the spontaneous reaction to happen? Discuss.